Life, magnified, shows off the wonders of the cell. Life is both wonderful and majestic, yet for all of its majesty, all organisms are composed of the fundamental unit of life, the cell. The cell is the simplest unit of matter that is alive. All living organisms, from the unicellular bacteria to multicellular animals, are products of repeated rounds of cell growth and division. Did you know that the human body is composed of trillions of cells, and each cell is an amazing world unto itself? It can take in nutrients, convert these nutrients into energy, carry out specialized functions, and reproduce as necessary. Even more amazing is that each cell stores its own set of instructions for carrying out each of these activities. So when we think about the fact that we started out as a single cell, would it really be amazing to know and understand the magic of how cells work? First, let's look at the main parts of a cell. The nucleus, generally considered the brain or the control center of the cell, contains all the genetic information necessary for the cell to function. Cytoplasm is a gel-like fluid part of the cell that contains the organelles and all the other components of the cell. And the cell membrane or the plasma membrane acts as a barrier which only allows selective movement of substances into or out of the cell. There are many kinds of cells in the human body, such as muscle cells, bone cells, skin cells, blood cells, and all with their own specialized function. Truly, so many remarkable biological systems working together to allow life, movement, cognitive function, growth, repair, reproduction, and so much more. The body's organization goes from cells to tissues to organ systems to a whole working organism. A cell reproduces by performing an orderly sequence of events in which it duplicates its contents and then divides into two. And this is what we call a cell cycle, which is a series of events that takes place in a cell as it grows and divides. So let's move on further to the amazing and magical world of the cell cycle. Cell cycle has three major stages: interphase, M phase or dividing phase, and cytokinesis. The M phase has two types, mitosis and the meiosis phase. The cell cycle is a complex system, but most cells of adult mammals spend about 24 hours in the resting stage that accounts for about 90% of the total time involved in cell division, known as the interphase, which includes G1 phase, S phase, and G2 phase. G1 phase, the cell grows and functions normally, wherein a high amount of protein synthesis occurs and the cell grows to about double its original size. Some cells at G1 phase enter a stage called G0 or quiescent stage, wherein cells are not able to move to the S phase but continue to function metabolically, but are not prepared for division. Examples are neurons, cardiac muscles, and red blood cells. S phase, where the genetic material such as DNA and chromosomes are synthesized and duplicated. DNA or deoxyribonucleic acid is a long molecule that contains our unique genetic code. G2 phase, in which the cell resumes its growth in preparation for cell division. Cell synthesizes its proteins and continues to grow in size. Cell double checks to make sure DNA is replicated and cell has enough organelles, and the cell continues to prepare for the next stage where it divides and reproduces in two ways: mitosis and meiosis. They are both important processes because without the two, cell division would not be able to occur. So let's move on to the next stage of cell cycle. Mitosis has an important role in the development of embryos as well as the growth and development of our bodies. It produces new cells that replace cells that are old, lost, or damaged. Technically, mitosis is a process where a single cell divides into two identical daughter cells. Mitosis begins with the prophase stage, where the chromatin condenses and thickens and are seen like rod-shaped structures. Condensed chromosomes consist of two sister chromatids joined at the centromere structure. At this stage is the formation of the spindle fibers and the disappearance of the nucleus and nuclear membrane. Towards the end of the prophase stage, the centrioles begin to move to the opposite poles and the start of the metaphase stage. During this stage, the rod-shaped double-stranded chromosomes line up at the equatorial plate. With the kinetochores attaching the chromosomes, the spindle is now fully formed. The next stage is the anaphase stage, wherein each pair of sister chromatids separate from each other. This happens when the chromosomes break at the centromeres and sister chromatids move to the opposite ends of the pole. Anaphase ensures that each daughter cell receives an identical set of chromosomes, and it is followed by the final stage of mitosis known as telophase. During this stage, the sister chromatids reach opposite poles, the chromosomes uncoil returning to its thread-like structure or loosely packed chromatin, and the spindle fibers disappear. The nucleolus and nuclear membrane reappear around each set of chromosomes. Cytokinesis is the division of cytoplasm. In animals, a cleavage furrow in the plasma membrane deepens and divides the cytoplasm into two identical daughter cells, each containing a nucleus. However, cytokinesis may or may not occur, but if it does not occur, it may lead to the development of cancer cells. Hence, mitosis stage is very important in ensuring that each daughter cell receives a complete set of chromosomes. After knowing the process involved in mitosis, let us now discover the meiosis stage. Remember that just like mitosis, meiosis also passes with interphase, G1, S, and G2 stages before they enter meiosis. The formation of sex cells is a central part of human reproduction, and these sex cells or gametes are sperm cells in males, egg cells in females. These sex cells are produced through a type of nuclear division that reduces chromosome number to half of that of the parent cell. And this reduction division stage is called the meiosis, that involves two successive cell divisions, meiosis 1 and meiosis 2. Let's look at this process in more detail. Meiosis 1 is the reduction stage where the chromosome number is reduced to half. Early in prophase 1, homologous chromosomes come together to form a synapsis, and an exchange of genetic material occurs in the process called crossing over. In metaphase 1, each pair of homologous chromosome attaches to a single spindle fiber and line up at the center. During anaphase 1, wherein the pair of chromosomes are then pulled apart by the meiotic spindle, which pulls one chromosome to one pole of the cell and the other chromosome to the opposite pole. In telophase 1 and cytokinesis, two new nuclei are formed, each containing only half the chromosome number of the original parent cell, and undergoes in the process of where cytoplasm divide. These two new cells undergo meiosis 2, which is the division stage and is similar to mitosis. In prophase 2 again, chromosomes condense, the nuclear envelope breaks down, and the spindle apparatus forms each double-stranded chromosome and attaches to a spindle fiber. The difference between prophase 1 and prophase 2 is that a daughter cell has only one copy of each homologous chromosome. Likewise, there is no synapsis of homologous chromosomes or crossing over. In metaphase 2, the chromosomes of each cell align at the equatorial plate, and the alignment is random since the sister chromatids are no longer identical, and there will be many different possible ways for these chromosomes to align. During anaphase 2, the sister chromatids are pulled apart as the microtubules shorten. The ends of the cell are also pushed further apart as microtubules elongate. In telophase 2, the nuclear membrane reforms and the cytoplasms are divided into 4 haploid daughter cells, and this division is called cytokinesis, which marks the end of meiosis, forming gametes or sex cells. But the question is, why do we still age over time despite the cell cycle and cell division process? As mentioned, our body is made up of cells, and these cells over the years are replaced at a slower pace. We age because our cells age. Our body is being worn down by the tiny cell processors because of oxygen, radiation, and our metabolism. Our body has so many mechanisms to repair this damage, but over time become less effective, and these changes affect the functioning of all body systems. Our bones and muscles weaken, our skin wrinkles, we lose our memory, our immune system weakens. Growing old means going through changes both mentally and physically. Aging is inescapable, nor we can sometimes avoid illness. However, we have the power to live a healthier life, and we have only one body in this life that we all want it to last as long as possible.